Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. As you know, these videos are intended for use by students who are enrolled in my microbiology class. If anyone else out there in YouTube land finds them helpful, by all means use them. Please hit like and let me know if they're helpful. That way I can continue to do these. If not, I'm wasting my time and yours. Anyway, also know that these are to provide, the intent of these videos is to provide a solid fundamental understanding of some of the major principles in microbiology for health science majors. This is not a research level course. And thirdly, if your instructor goes into more detail or covers it differently, please learn what your instructor wants to know. This is what I like to cover to provide the foundation for my students. Now, we have been talking about the immune system in the past few videos. We've talked about the innate or non-specific immune system, the one we're born with, where all of our phagocytes attack foreign cells. We learned about the different types of white blood cells, and there was 4 major ones that we talked about: neutrophils, eosinophils, basophils, and monocytes, and the role that they play. We talked about specific immunity in another video, what we call adaptive or acquired immunity, and we acquire that immunity after exposure to some new antigen that our body's never seen before, or our body adapts to this new antigen. And once we developed antibodies against that antigen, those antibodies are specific for that one antigen. If a new antigen comes in, that antibody won't recognize it. Another B cell that's never been exposed, called a naive B cell, can develop antibodies against that new specific antigen. And so I can have B cells making antibodies against all these different specific antigens. Each B cell and set of antibodies is dedicated to that specific antigen. We're going to talk a little bit more about that. We're going to finish up some of our adaptive immunity. So, just sort of a quick review. As you guys know, when one of our B cells, so here's a B lymphocyte that has some antibodies on the surface of it. We're going to talk about which antibodies are usually on the surface of these, okay? We're going to go through the different classes of antibodies, because there's really 5 classes of antibodies that we could talk about. The ones that are actually on the surface of B cells are called the IgD antibodies, or immunoglobulin D. We'll come back to that. But let's say this immune cell comes in contact with, say, some virus or bacterium that has an antigen on its surface that binds, and the B cell can interact with that, and a T helper cell. And the T helper cell can release some chemicals called cytokines that activate the B cell. Regardless of those details, which we've talked about in a previous video, what happens when these B cells encounter that antigen and are stimulated by cytokines is this B cell will start to clone itself rapidly and make lots more of itself. Okay? So we'll have all these B cells, and because they are direct clones, they are genetically identical, then this antibody that's on the surface is the same as that antibody. They would all recognize the same antigen. And so as the B cells start to rapidly multiply and make more of themselves, essentially what happens is some of the B cells will remain as what we call B memory cells. I'm going to put a B with a little M there. And these cells will hang around for a long time, sometimes weeks, months, years, sometimes a lifetime, and they have that antibody against that specific antigen. So if you have a second or third or fourth exposures in the future, when that same bug, whether it's a bacterium or virus, comes in and has this same antigen on its surface, these cells will recognize it much more quickly and have a much more rapid response. So on first exposure, it usually takes a couple of weeks to get to this level. But on second exposure, we can skip some steps, and this cell will start to clone itself rapidly and have a much more rapid immune response and neutralize or kill, result in the killing of the invader much more rapidly than it initially took. That's why the first time you get exposed to something, you might get sick. The second and third and fourth time, you may not. Different illnesses work differently, but nonetheless. So the B memory cells hang around and can recognize this foreign invader and then divide rapidly, cloning themselves, making more memory cells, but also making more of these cells. And these cells will continue to divide, but they become big, angry, activated B cells that we call plasma cells. Plasma cells will actually make these antibodies and then secrete them. And because we have such large numbers of these B cells, these plasma cells, then the amount of antibodies that can flood our blood or a specific tissue is massive. And some of these B cells live in lymph nodes. And when your lymph nodes get swollen, that's a sign some infectious agent has come in, like when your tonsils get swollen. And they're massively secreting tons of these antibodies and flooding our blood, our fluids, our humors with this humoral response, this antibody-mediated or humoral-mediated immune response. Now, when it comes to this antibody, before I get into the different types, some details I want to explain. And I know I've gone over this before, but it's important to hear this. When I look at these antibody proteins, they're usually some Y-shaped little proteins. Now, as you guys know, proteins are long chains of amino acids. So if I take a whole bunch of amino acids and I string them together with some chemical bonds between them, then I get what's called the primary structure of the protein. Those proteins can then fold up and take on three-dimensional shapes by having some internal chemical bonds. Well, it turns out that antibodies have these protein chains that fold up a certain way. And they're made up of 4 separate proteins that are held together with some chemical bonds. 2 of the proteins are these big funky hockey stick shaped proteins. And then 2 of the proteins are similar, but they're shorter. Okay? These 2 proteins are almost identical. And then these 2 smaller proteins are basically identical. They're held together with some chemical bonds called disulfide bonds. Those are bonds between 2 sulfur molecules, that sulfur atoms that form what we call a disulfide bond or disulfide bridge. Now, that holds these 4 proteins together. The 2 ones that are in blue are called the heavy chains. And they're called heavy chains because they are chain-like proteins that are all linked together, and they're very large, and therefore their molecular weight, when we do what's called the molecular weight of a protein, how much does it weigh? They're much heavier than the 2 smaller ones. The 2 smaller ones are referred to as the light chain proteins because they're much lighter in weight, okay? They're smaller proteins. Now, we're not researchers, so we don't care about the number of kilodaltons and how they weigh. We just know the bigger ones are the heavy chains, the smaller ones are the light chains. Now, both the heavy and the light chain proteins, as you guys should know from early in the semester and other biology classes, when I look inside the nucleus of a cell and I see a chromosome, a long piece of DNA, all the bases on this chromosome are the instructions on how to code for a protein. So let's say this whole series of bases on this chain is the instructions for which amino acid goes first, goes second, goes third to make our protein. Well, the gene for these proteins is mostly what we call constant, in the sense that if I look at the amino acid sequence for this part, between almost any antibody in one person's body, that part of the amino acid sequence is unchanged, or it's constant. So what we refer to as part of the molecule we call the constant region of an antibody. It is unchanged in its amino acid sequence. Their amino acid sequence is pretty much the same no matter what. But there is an area here, right at the tips of this Y-shaped molecule, that is called the variable region. And there's some thought that this is the reason it takes a while for an immune response at the humoral level, the specific immunity, to occur. Now, the variable region, the reason it's called that is because the amino acid sequence varies or is different. And the reason it's different is because it's that area that will bind to a specific antigen. So let's say, for example, I expose these white blood cells to a foreign protein on the surface of a cell. So I have a foreign cell that enters the body. Maybe it's a bacterium. And on the surface of that bacterium, I have all these different proteins that are different shapes. And remember, there's more than one of each of these proteins. There's thousands of them on the surface of a bacterium. Each one of those proteins could be recognized by our immune cells if they're a protein that is not similar to one from our body. You have to remember the bacterium E. coli that lives in your gut has about almost 60%, around 56% homology, meaning the genes are identical to human genes. They have the same proteins. So some of these proteins might be the same proteins that are on the surface of our cells. And so our immune cells wouldn't recognize them, because if they did, they would also recognize our cells as foreign cells and attack them. And there's another video we'll talk about called autoimmune disorders, where your immune system attacks your own cells. It kind of sees our own cells as foreign and attacks them. That's a whole other topic. But let's say a particular B cell bumps into this foreign cell. It's eventually going to make an antibody that will bind to maybe that specific antigen on its variable region. And this blue marker doesn't work very well. So here's my little antibody, and here's its variable region, and it's got its little chains. And so another one of this exact same antibody can bind to this antigen. So I can have multiple antibodies binding to the same foreign cell. But here's the other cool thing. Because there are two variable regions, if there's another of the exact same foreign cell with the exact same protein on its surface, then I can actually bind up two of these foreign cells. Because these antibodies will recognize anything that has that protein on its surface. So I'm going to draw a little bit more simplified. Instead of the four chains, I'm just going to draw more like the little Y. But now another antibody will bind here, and another one can bind here. And this variable region is unique to that foreign protein. And essentially what happens is, because they have more than one variable region, then one antibody can bind two of those antigens, or two cells that have that antigen on its surface. And then we can get what's called an agglutination reaction, or a clumping reaction. We can clump all the bad guys together. We've talked about that before. It makes it to where the other white blood cells, when they arrive, can eliminate all of them at once, or a whole bunch of them. So when we look at the structure of an antibody, they are Y-shaped proteins. They have four proteins involved in their complex. Two heavy chains, the larger ones, two light chains held together by disulfide bonds. The constant region, the amino acid sequence never changes. From one antibody to the next, it's pretty much the same. But when my body encounters my immune system encounters a foreign cell that has a specific antigen on its surface, then a naive B cell can make an antibody and rearrange the DNA, the genetic sequence of the variable region, and alter the amino acid sequence so that it will bind to that specific antigen. And what could happen is this. Let's say I have a different foreign cell come into my body. And that foreign cell does not have this same antigen. It's got a different antigen. I'm just going to use the letter Q. Okay? And a whole bunch of these come in. And they have a different foreign antigen on their surface. Then this B cell secreting these antibodies will not recognize it. But if I had a different naive B cell that can start making antibodies against the Q protein, this is why we call it specific immunity or acquired, is because only after exposure to that foreign cell will some B cells recognize it and make antibodies. And then those antibodies will start to bind to these proteins on their surface and clump them together. Excuse me. So now I have a whole class of B plasma cells that can make antibodies against one foreign antigen, and another group that can make antibodies against a different foreign antigen. So every time we get exposed, our body can have B cells that are naive become activated and become dedicated plasma cells that will make antibodies against a specific foreign antigen. And they're always going to make some memory cells that will remember it, so that once we wipe it out, if it tries to reinvade the body, we'll get a much more rapid second response, third response, and wipe it out before it causes the illness in most instances. Okay? This is why if you get chicken pox as a kid, you don't get it again, because your immune system can recognize it. So, I want you to understand that when B cells encounter a foreign antigen, they can undergo rapid cloning of themselves. Some become memory cells, some become plasma cells that secrete antibodies. The antibodies are made up of 4 chains, 2 heavy, 2 light. The constant region is unchanged, and this is where our cells could bind to them. For example, a phagocyte coming by here, like a macrophage, will recognize this region. The constant region is sometimes called the effector region, and the other white blood cells will bind to that and then engulf this. That's how they recognize antibody-labeled antigens. Now, the variable region is the part where the amino acid sequence can change and become unique to binding to a specific foreign antigen. Now, one of the things, I'm going to erase some of this. Well, one of the things that we can do, and we're going to talk about in another video, is how do we develop vaccines? How do vaccines work? And one way that vaccines can work is that I can give you a virus that has had its DNA or RNA destroyed so that it can't replicate. It can't do any harm to you, but you get exposed to part of the virus, you make antibodies against it, so that when the real virus comes in and tries to infect you, your body will wipe it out before you even get the infection. That's kind of how vaccines or immunizations work. In some instances, by the way, we don't even give you the whole virus. We can just give you a part of the virus that has the antigen on it, the protein, or in some instances, we can simply inject the protein, and we can make antibodies against the protein, so that when the real cell enters your body, or virus, and has that protein on its surface, you already have antibodies against it and can eliminate it or neutralize it before it causes you to get ill. That's kind of how vaccines work, okay? So, anyway, now, what I want to talk about, now that we understand the structure of antibodies, and that they're part of the antibody-mediated or humoral-mediated immune response, the specific immune response, I want to talk about the different classes of them. And there's 5 classes of antibodies that we can talk about. And it's really more complex than this. And antibodies are also called immunoglobulins. Globule means to clump, and immuno, they're part of our immune system, and they will cause clumping or globular reactions. They'll form globs of stuff. And so they are abbreviated as Ig sometimes. So when we look at the immunoglobulins, the 5 classes of antibodies, one class is referred to as IgA. Okay? I'm just going to go in alphabetical order, and I'm going to give you the details that you need to know about each one of these. So when it comes to IgA, that marker's getting a little bit weak. What I want you to know about IgA is very often these form what we call dimers. Di means 2, and mer means unit. So when these antibodies are formed, I'm not going to draw all 4 chains, but suffice it to say that when these antibodies are formed, the effector region or the constant region of one binds to the constant region of another, and then they can have 4 variable regions for a specific immunoglobulin. So IgA forms dimers, 2 antibodies stuck together. Now, These are the second most numerous of the antibodies that are secreted. There's a bunch of these, okay? The most numerous is going to be IgG, but we're going to talk about those in a moment. They are found in body secretions. So, saliva can have IgA antibodies in it. Tears, mucus, sweat, breast milk. IgA can be passed from mom to the baby through breastfeeding and therefore provide some immunity against specific pathogens. So we call that passive acquired immunity. Actively acquired immunity is immunity that is acquired that your body actively makes the antibodies against it because you've been exposed. In passively acquired immunity, the immune cells are given to you passively by someone else who's already developed them. A lot of times through breast milk, or we take advantage of it now, like for example, if someone has had COVID or Ebola virus, they've been exposed and they have the antibodies, we can actually take some of their blood, isolate the antibodies, and then inject those antibodies into someone else, providing them that immunity. Although it's only temporary, it's effective. So, anyway, IgA forms dimers. It's the second most numerous, and they are found in a lot of body secretions. And we usually find them like in the urinary and reproductive tract, or urinary and reproductive secretions, the respiratory tract, the GI tract. So they protect a lot of our mucous membranes internally so that when we take things in, these antibodies can neutralize it rapidly. So once developed, IgA can be found in a lot of our secretions. Saliva, tears, mucus, sweat, even breast milk, and passed into babies that way. It does not cross, by the way, the placenta. It's passed through breast milk. That's passive immunity. Okay? So there's IgA. The second one I want to talk about is IgD. Okay? Immunoglobulin D. What we know about immunoglobulin D, it is probably the least numerous in serum and fluids. We don't find this one floating around in body fluids. They are bound to the surface of B cells. So the IgDs, their effector or constant region is embedded in the cell membrane of B cells. And then it is the variable region that will allow them to recognize other antigens. Something that we do know is that they can activate basophils to respond and mast cells. So if there are any IgD antibodies in fluids, they can activate basophils and mast cells to respond to that area. They're probably the least understood, so we don't know a whole lot about them as much as we do as the others. So now I'm going to erase IgA because I'm running out of board space, and I'm going to add the next one, which is called IgE, or immunoglobulin E. By the way, IgD tends to be on the surface of cells, or it's usually a monomer. It's only IgA that forms dimers. If we were to talk about IgE, or immunoglobulin E, there's a few things to know about immunoglobulin E. Let me find my good bookmark. Okay. So for IgE, they are on the surface of basophils and mast cells. They can stimulate these cells to release histamine. So They are elevated in allergic responses or very often parasitic infections. So these are on the surface of basophils and mast cells. If a basophil with IgE on its surface encounters an antigen, it can release histamine, triggering an allergic response, or alerting our body that there's a parasitic infection. Okay? So they tend to be elevated in allergic responses and parasitic infections. Or at least the histamine is causing an inflammatory response. I think that's about all I want you to know about IgE. Now, the next one is called IgG, or immunoglobulin G. Okay. A few things to know about immunoglobulin G. This is the most numerous in body fluids. There's tons of IgG in body fluids. They can bind to antigens. And coat the cells that have that antigen on them. Okay? So, the effector region is recognized by phagocytes. Well, particular phagocytes that recognize them are basophils. Did I say that right? No. It's not basophils. My bad. It's macrophages and natural killer T cells. So they can trigger whatever they bind to be phagocytosed or destroyed. So they're part of well, they're usually some of the ones that are coding some of these foreign invaders and are recognized. It is the effector or the constant region that some of our macrophages and natural killer cells can bind to, and the macrophages will engulf and phagocytose the foreign invader. The natural killer cells will release the perforins that will poke holes in the foreign invader and kill it. And they are also released as a monomer, single units. Again, the only one that's a dimer is IgA. Now the last one I want to talk about is IgM. And you can think of the M as standing for massive. So when we talk about IgM, or immunoglobulin M, they form what we call a pentamer. So it's 5 units from pentagon, or penta means 5. So there's 5 units. So essentially what happens is the constant region of these will interact with the constant region of 4 others. And so I have 1, 2, 3, 4, 5 of these IgM molecules all kind of stuck together, and then the variable regions out here will all recognize the same surface antigen. Okay? And what that does is it allows them to clump together massive amounts of the foreign invader. And we call that agglutination, or sometimes it's called flocculation. It becomes flocculent. Okay? If they do exist on them as a monomer, they're on the surface of B cells. But usually in the fluids and the humors, they are released as pentamers, and they can clump together large amounts of these foreign cells. So, and only imagine now if I had another pentamer here that was bound to this cell, then I could get 10 more cells bound up. And then another collection could do that, and you can really clump up tons and tons of cells that cause massive agglutination reactions, and then our other phagocytes can come in and wipe out all of this stuff. So that's what I want you to know about IgM. Okay. That's really it. I don't want to go into too much more detail than that. So you should know that antibodies are secreted by plasma cells, or they're on the surface of certain B cells. The 5 classes, you should know the 2 heavy chains and 2 light chains. You should know the constant region doesn't change. The variable region is what allows them to recognize very specific antigens. And those antigens can be free-floating, like exotoxins, or they can be on the surface of foreign cells and viruses. And then know the 5 different types. IgA is the second most numerous. It exists as a dimer. IgD is the least understood. IgE, they're also monomers. And for IgD, they're on the surface of B cells. For IgE, they stimulate mast cells and basophils to respond in allergies and parasitic infections. IgG is the most numerous in our body fluids. It can bind to antigens and coat the cells, alerting other white blood cells that they're to come here and erase these guys, and they stimulate phagocytosis, or the elimination or killing of the foreign invader by natural killer cells. And then IgMs are massive in that they form a pentamer, and they can cause agglutination reactions, large clumping of foreign antigens or cells with antigens on their surface to clump them together, and then makes it easier for our other immune cells. By the way, IgMs are also very often the ones that are the first responders. They're usually the ones that are going to react first with things because there's so many of them. I think that's all the stuff I want to tell you about them. So I believe that we will we'll conclude this video with that information. It's not a lot of information, but it's important information that you should know. Anyway, I hope you learned something. I hope you found it helpful. I hope you had as much fun as I did. And I hope to see you in the next video. Thanks for watching.